Hello, welcome to Biotouring Cell Trait webinar. I am Jenny, Single Cell Analytics Marketing Director of Biotouring, and thank you very much for being here with us today for this webinar on Similarity Weighted Non-Negative Embedding. To keep our meeting going well, here are some general guidelines that you should note. The meeting is being recorded and will be made publicly available after the webinar. All the attendees are muted. We will have 15 minutes for a Q&A section after the presenter has finished his talk. And if you have any questions along the way, please leave it in the message box. For PC users, the message box is located below the screen. If you use a mobile device, then please tap Participants, then go to Chats, and you can then leave a message there. In the Q&A section, if you want to directly speak with the presenter, then please raise your hand. If you're on a PC, just click Participants and click Raise Hand on the right panel. For mobile devices, please tap More and Raise Hand. Now, I'm very happy to introduce today's speaker, Yan Wu. Yan is working on applying computational and experimental approaches to understand the processes underlying human development at single-cell resolution, and he has previously developed computational approaches for visualizing single-cell datasets and for mapping the cell types, resulting from single-cell overexpression screens. Yan is most recently working on mapping the DNA accessibility landscape of human brain development. Okay, now I will quickly go through the agenda. First, Yan will deliver his talk on his algorithm SWNE, and later we will have a 15-minute Q&A section. Okay, so now I'm going to pass the mic to Yan.
Oops. Can everyone hear me?
Yeah, I can hear you well.
Okay, perfect. Hey everyone, so today I'm going to be going through a method that we developed for visualizing single-cell datasets called Similarity Weighted Non-Negative Embedding. And I'm just going to take you through some of the, ideas behind why we developed this, and then demonstrate it on a couple examples and kind of compare it against some of the other visualization methods out there. So one of the kind of major breakthroughs in the past couple of years has been the development of these high-throughput single-cell RNA-seq and chromatin accessibility methods that have largely been driven by advances in both droplet microfluidics and another kind of cell isolation method called combinatorial indexing. And without going into too much detail into these two methods, they've really enabled the characterization of novel cell types and development one of the first types of visualizations, or analyses that most people do, is try and visualize these data sets to generate sort of a 2D map and a summary. So we go from these, data sets are typically thousands or tens of thousands of genes by, however many cells you have, or even hundreds of thousands of accessible sites. And we want to kind of compress that down into 2 dimensions so that, a reader or a reviewer can kind of get an understanding of what the data set is. But the issue is that, so for example, we here have a t-SNE map on Hematopoiesis cell types, and this is from a 10x genomics data set. So you can kind of see this is a layout of all the different cell types in the data set. We can see the monocytes and the T cells and the NK cells and this, the B cells. We can see a little bit about how far away they are from each other and how many cells are in each cluster. So kind of an example of how we would summarize one of these data sets. But one of the issues with t-SNE, and t-SNE is one of the most popular visualization methods out there, is that it can distort data sets. And kind of most notably, it distorts distances between clusters and cell types. So on the left, we have kind of a bunch of points in their original coordinates. And on the right, you can see what happens after we run t-SNE. So the fact that the blue and yellow clusters were closer together kind of becomes destroyed through this process. So just to formalize this idea a little bit more, for these visualizations, we like to think about it in terms of preserving global and local structure. So one example is on the left is an example of good local structure. So we have a bunch of cities, right, in the US and in Asia. And this visualization on the left is able to resolve distances between close cities. So we can see that Boston and New York are distinct, and Beijing and Shanghai are distinct. But it distorts the relative distances between those cities. So for example, on the left, you can see that Boston, LA, and New York look equidistant when in fact they're very much not. And Boston and Beijing are not as far away from each other as they should be. So on the right, we have an example of good global structure, but poor local structure. So now we have maybe more accurate relative distances between faraway cities, but we can't resolve necessarily the distances between close cities now. Now Boston and New York look like they're on top of each other, and Beijing and Shanghai look like they're on top of each other. So a second kind of challenge with a lot of these visualizations, especially when you're still kind of exploring your data set, is that they lack context. So this is the same Hematopoiesis data set, except now we've taken away all the cluster labels and replaced them with numbers. And this is something similar to what you might get if you were kind of generating one of these plots for the first time. And while it does tell you how many clusters you have, how big the cell types are, or how many cells are in each cell type approximately, it doesn't really tell you a whole lot biologically, right? Like these could be literally anything. So our kind of way of thinking about this is, what if we added landmarks to facilitate interpretation? So now we have the same example with cities that we have before, but we've removed all the kind of city names and replaced them with city IDs. And we don't know what cities these are right now, but if we have landmarks like the Statue of Liberty or the Forbidden Palace or other kind of landmarks, we can start to identify these cities. So we can start to say, okay, city 3 is probably New York. And that also tells us a little bit about kind of the neighboring cities. So if city 3 is New York, then city 1 and city 2 are probably in the US more. So the idea of adding these landmarks, I think, is something that we're really, really interested in. So now to go into a little bit about how SWNE actually works. So the first step, it starts with a matrix factorization. And we take this big matrix and we break it down into two much smaller matrices. And this sort of is essentially running that first step of dimensional reduction and generating a sort of a summary of the data set. And this lower dimensional space will eventually act as a skeleton for the visualization. So each of the factors in that lower dimensional space, we take and we project into 2 dimensions using pairwise similarities. And we take all of the cells and all of the genes, because again, we have this genes by factors matrix and the cells by factors matrix, and we project them relative to the factors. So again, the factors are acting like this skeleton, and then we can visualize the cells and any genes we might want to use as landmarks on top of that skeleton. So the final step, and one that's kind of critical to the actual performance of SWNE, is to use a nearest neighbors network to smooth cell embeddings. So this essentially forces SWNE to be a little bit more like t-SNE, and it pulls cells that are close in the original gene expression space apart together in the visualization space. So going through SWNE, we're gonna go through and benchmark SWNE on a hematopoiesis data set. And this is a data set from a 2015 Cell paper where they looked at single cell mouse hematopoiesis trajectories. And again, hematopoiesis is kind of the generation of these blood cells. So starting with the MPEP are the progenitors, and it kind of goes down along two branches. So on the right, you'll see ERY, and those are the erythrocytes, so the white blood cells. On the left, you'll see all these things like neutrophils or monocyte progenitors, and this is kind of the monocyte lymphoid branch. So this is an SWNE visualization of this data set from, and this is also a figure in the paper that we've published alongside SWNE, so you can take a look there if you want. But basically, we can see that SWNE is really able to capture both the monocyte and erythrocyte trajectory, and this is without assuming that the data set is continuous in any way. This is just kind of taking this data and putting it into SWNE. So again, we can see starting from the green kind of progenitors, we can branch out towards these erythrocyte cells or these monocyte or lymphoid cells. So again, this idea of adding landmarks we think is really, really useful. So now we've added these kind of marker genes in red, and then also we've gone ahead and interpreted some of the factors, which often kind of correspond to these gene sets, and they're in purple. So just going through real quick, so we can see ApoE is a marker for progenitor, the kind of progenitor-like state in these mouse hematopoiesis cells. You can see it's visually very close to these green cells. And then on kind of the top left branch, you can see that SWNE2 is the marker gene is very spatially close to the neutrophils, which it is primarily expressed in. And for erythrocytes, you can see, I think it's metallothionein 2. It's a metal binding protein, is primarily expressed in erythrocytes, and it's also visually very close. So you can imagine that, if you didn't have these cell type labels, having these marker genes kind of embedded on the visualization would be very helpful for that first step of interpretation. And we think it's also pretty helpful for kind of a final plot as well, in terms of really emphasizing, which genes are expressed in this data set, which genes mark each of these clusters. And we can take a quick look at these factors. So with these factors, you kind of have to go in and interpret them yourself, much like you would an unknown cluster, but they can be very helpful depending on the type of data set you have. So in this case, we got a factor inflammation that comes out really strongly, and again, that's spatially very close to the neutrophils, which is the primary function of these neutrophils. So now we want to visually compare SWNE, t-SNE, and UMAP. And UMAP is a newer visualization method that actually works really well in a lot of cases and addresses some of the issues of t-SNE. So we want to go through and compare all three, and we can see that SWNE is really able to sort of compress the data set onto its primary axes of variability. So with t-SNE, you can sort of see the branching structure. And the UMAP, it's kind of the same thing, but the erythroid and the monocyte branches are sort of flattened out. They're very, very spread out, whereas in SWNE, they're really kind of condensed onto that primary axis of differentiation. So we really think that, especially for this data set, SWNE is able to perform really, really well compared to these two other methods. So this is just a quick visualization of differentiation pseudotime, which again sort of shows the same thing, right? Where SWNE is really able to capture that gradient of differentiation, whereas t-SNE and UMAP, it's kind of spread out, and a lot of the kind of area of the plot is devoted to either highly differentiated cells or progenitors, and it doesn't really show as much of a gradient. So just to kind of formally quantify some of these things, we went through and did a quantitative evaluation of these embeddings. And just really quickly, to quantify global structure, what we did is we took all the cell types in these data sets, and we sort of calculated all pairwise distances in both the original space and the 2D visualization space, and we took a correlation. And similarly for local structure, what we did is in the original, a couple thousand gene expression space, we took a look at, for each cell, what are all of its neighbors, and we saw how that compared with the neighbors in the visualization space, you can see that, all three methods do pretty well in terms of, capturing the same neighborhood, capturing that local structure, though t-SNE, as you might expect, does extremely well. But SWNE does really well in this case of capturing global structure. And UMAP doesn't do so well. It is a little surprising because we expect UMAP to be at least far better than t-SNE. But if anyone has any questions about that, I can go into some thoughts we had about that later. And it might have something to do with the fact that UMAP doesn't do quite as well on these smaller data sets. So now we want to try and take a look at how SWNE works on a larger data set, especially one that has discrete cell types. So the past hematopoiesis example is an example of these continuous differentiation trajectories. So here we can see SWNE, t-SNE, and UMAP. And just going through kind of quickly, you can see that, for SWNE and UMAP, there's a decent amount of white space in between some of these clusters, whereas again, t-SNE sort of compresses all the distances between clusters, so it takes up a lot of the plot, but doesn't really, accurately maybe represent the orientations of these clusters. And just going through an example of what that means. So if you take a look at the red box for t-SNE, we have sort of 4 clusters in there, and we have these 2 interneuron clusters, IN7-8 and IN6, and then we have these 2 oligodendrocyte clusters. So we have oligodendrocytes, which is OLI, and then OPCs, which are oligodendrocyte progenitors. And they're all really, really close to each other, and they're all basically kind of equidistant. Whereas you see in SWNE and UMAP that the oligodendrocytes should be pretty far from the interneurons, and vice versa, because they're pretty different cell types. So this is kind of an example of distorting distances, relative distances between clusters. Now, obviously, this becomes challenging for any visualization when you have this many clusters, but kind of SWNE and UMAP, I think, try their hardest to try and orient these clusters correctly, whereas t-SNE really only cares about sort of that cluster separation, that local structure. So just to go into another example of how landmark gene embedding can add biological context in this case, just pointing out kind of 2 marker genes, CBLN2, I believe is a layer 2-3 excitatory neuron gene, so that is spatially very close to the EXL23 cluster, the layer 2-3 cells. And then over here on the bottom left, we see that SLC1A2 is marking these astrocytes, and it's right on top of these cells. We can tell that these cells are expressing this marker gene from the context. So moving on, we kind of wanted to try and benchmark SWNE on a large continuous trajectory data set. And because again, there's the possibility that UMAP struggles with these kind of smaller data sets because it's just not, the parameters might not be optimized for data sets less than 5,000 or 10,000 cells. And we see here that, again, SWNE and UMAP seem to really be able to capture the trajectory structure of this data set, while t-SNE really flattens everything. And again, SWNE has these marker genes that are able to mark these macrophages with FCGR1 and these B cells with MS4A1. And looking quickly at the quantitative evaluation, we can see that SWNE and UMAP perform pretty similarly, while t-SNE does much better in terms of local structure, but doesn't really capture the global structure, those distances between clusters. So now, kind of switching gears, we wanted to also apply SWNE to chromatin accessibility. So for kind of RNA-seq, single-cell RNA-seq, we are using a method for matrix factorization called non-negative matrix factorization. And for chromatin accessibility, you start out with this really large, it's actually a binary matrix. So another method called latent Dirichlet allocation tends to work really, really well in this case. And it's implemented in a package by Gonzalez-Blas et al. Called SysTopic, which is a really great package if anyone does this type of data analysis. And after kind of decomposing the matrix in the same way, instead of trying to identify or trying to embed individual genomic sites, which would not be that useful for interpretation, we actually created functions so that if you're using SWNE with this type of chromatin accessibility data, you can embed the promoter accessibility of genes as well as the binding site accessibility of transcription factors. So just going through an example with promoters. So this is applying SWNE to chromatin accessibility of a chromatin accessibility map of the human brain. So on the left, we have these excitatory neurons. On the right, we have these kind of oligodendrocyte cells and some microglia, and then we got some interneurons on the bottom, endothelial cells in purple. And we can again see that how SWNE is really able to try and capture the distances between these clusters. It doesn't just kind of compress everything into the same distance. And it also is able to embed the promoter accessibility of a lot of these key marker genes. So CUX2 for excitatory neurons, again, the same gene that we kind of pointed out before, SLC1A2 for these astrocytes, and then GAD2 for these interneurons. So instead of looking at gene expression, now we're looking at the accessibility of the gene promoters. And this is kind of a second example of applying SWNE to chromatin accessibility, where we're going kind of back to this hematopoiesis data set, starting with progenitors in kind of the bottom right, and then moving along the two branches, the lymphoid monocyte branch on the left, and then the erythrocyte branch on the right. And in this case, we're kind of demonstrating the ability to embed both promoters. We can see that with the genes that have a kind of -PR at the end, but we can also embed the binding sites for these two transcription factors. So CEBPB is a, I believe, monocyte marker, and we can see that both its promoter accessibility and transcription and binding site accessibility really line up with the monocyte cells. And same for GATA1 and these kind of erythrocyte progenitors. We can see that GATA1, both the promoter and the transcription factor binding site, are marking this erythrocyte branch. So just to kind of summarize, we can run SWNE on chromatin accessibility as well, and we just kind of replace the embedding of genes with the embedding of promoters and or binding sites. So just a quick final example, SWNE can also be applied to text mining and other high-dimensional data sets. So this is a data set of, I think, 20,000 or so news articles where we just, for each news article, we created what's called a word vector, which is a vector of all possible words and kind of across all these data sets. And we just, count the number of words that occur in each data set. And, we can see some separation, which was interesting. And then this kind of landmark embedding now in this case is applied to this dictionary of words, and we can see different words mark out each topic. So you can see that a lot of the computer, I guess, articles mention Windows and Apple a lot, and a lot of these science articles and kind of the yellow, they tend to mention intellect a lot. So, just an example of how SWNE can be used for other applications as well. So in conclusion, SWNE is able to balance capturing both global and local structure. It outperforms t-SNE according to a lot of these benchmarks, and performs similarly to UMAP, at least according to what we've seen. And just a quick note on UMAP, UMAP we think is a great visualization, and a lot of what we do now is use both SWNE and UMAP to visualize the same data sets, although with different kind of use cases. So SWNE also enables the embedding of landmark genes to kind of facilitate interpretation, to really add that biological context. And again, it can interrogate TF transcription factor binding and accessibility dynamics, and can be applied across a wide variety of data sets. So yeah, we have our GitHub page, and if you have any questions, you can feel free to email me or leave a comment on the GitHub page. And we also, this work has all been published in Cell Systems, if anyone is interested. So yeah, I guess I'll take any questions now.
Okay, thank you, Yan, for your valuable sharing. And now it's time for the Q&A section. So I just received some messages from you saying that the sound is very weak, and it seems that you cannot capture the first part. But I would like to repeat again that if you have any questions, you can leave your questions in the chat box, or if you want to directly ask Yan some question, then please raise your hand. We will unmute you and you will be able to chat with Yan. All right? So yeah, got one question. Yan, can you see the box?
Yeah, I think I see a question there. So I think Nan asked, how fast is SWNE compared to t-SNE and UMAP? So we find that SWNE is comparable to t-SNE and UMAP. It's I forget exactly what the numbers are. We do have a benchmark in the paper discussion, as someone helpfully mentioned. And the one thing we'll say about SWNE is you do need to do some sort of feature selection before you run it. Otherwise, if you try to run it with the full kind of 20,000 genes, it does become sort of slower. But yeah, otherwise we feel that it's just about as fast as t-SNE and UMAP, at least as implemented on SWROT. And yeah, I think, so just to go through the questions one at a time, I think there's a question on implementing SWNE and ScanPy. And yeah, we'd love to have a Python implementation of SWNE. If you go to the GitHub page, you'll see that it's on R right now. But yeah, it's definitely something we're looking to do. I think Tan is asking, how do you find the most valuable landmark genes to put on the 2D plot? So I'll just go through a slide here that sort of addresses that. So the landmark genes that you probably want to find are genes that you should probably, it's a great question. So those genes are probably genes that you should find either from a literature curation or sort of a, doing differential expression under clusters. So we provide a couple functions that will kind of help check any genes you have to see if they're going to be good genes for landmark genes, because you can imagine if you embed a non-differentially expressed gene, it's not going to be very useful. And here's just kind of a plot showing that. So we just basically plot the maximum log-fold change in the gene expression data set versus the maximum log-fold change sort of in this 2D space. And any gene that is kind of, in the top right quadrant of this plot is probably going to be a pretty good candidate for embedding. And just to kind of go over the idea of these landmark genes again. So if we look at, these two types of gene embedding, so we basically took all human genes and embedded them on the Hematopoiesis data set and just built a heat map of where they fell. So if you take the differentially expressed genes, and those are the genes that are in the top right quadrant of that plot that we showed before, they tend to fall on the edges, and they tend to, as you can see, mark the cell types, whereas the non-differentially expressed genes just tend to fall in the middle of the plot. So if you have any genes that are like right smack dab in the middle, then those will probably be not good landmark genes. So Jan is asking, is it possible to get a list of genes for each cluster or region? Yes, that's kind of theoretically possible. We unfortunately haven't implemented that functionality yet, but that's definitely something I'll try and do. So the idea would just be kind of, each gene marks out a physical space, and you could kind of draw a circle around some physical space and find the genes that are there, which I think would be really useful and something that we're definitely trying to do, but it's not quite there yet.
Yeah. Any other questions? If you have questions and want to talk directly with Yan, then please raise your hand. We can unmute you. Any other questions so far? There's one question from Tan.
Okay.
Question, yeah.
Yeah, so that's a great question. So I'll just kind of go back to to this slide. So the question was, describing more in detail how to embed both genes and cells on the same plot from TAN. And I'll just go through that one more time for everyone's sake. So the idea is that when we kind of run this matrix factorization, we get this lower dimensional space, right? And this is really common for single cell RNA-seq analysis because, you know, even though we're measuring 20,000 genes, a lot of those genes are highly correlated with each other in that there aren't 20,000 different, you know, kind of things going on in the cells. They're usually only going about like 20 to maybe 40 dimensions of lower dimensional space. So after doing that matrix we do this matrix decomposition, we get, let's say, a matrix of genes, let's say like a couple thousand by 20, and then a matrix of cells, so cells by, let's say, 20 factors. For each of these factors, we can take them and put the factors on a 2D space. So we do that using pairwise similarities from this matrix. We just calculate how correlated each of the factors are in this matrix. And if they're highly correlated, we put them together. If they're not, we put them further apart. And that's using a method called salmon mapping. But the fact that these factors are now on this 2D plot means that we can go back. Because this is a genes by factors matrix, we can put any genes that are in this matrix on the plot as well. So we can put the cells on the 2D visualization using the scores from this matrix and the genes from the loadings matrix. And the way that's done is that it's all just kind of relative. So if, let's say, this cell has, or this example cell has a really high score for factor 1, but is 0 for factors 2 and 3, then it'd basically be right on top of factor 1. And it's the same concept for the gene. If this gene has a really high loading for factor 2, then it'll be really close to factor 2 and then further away from the other factors. So it's just sort of putting both the genes and the cells on the same visualization relative to the factors.
Okay, does that answer your question, Tan? I can unmute you so that you can directly talk to Yan.
Yeah, thanks for your answer. One more question is the coordinate of the SWNE1 and SWNE2 is interpretable? Because it's quite hard to interpret the coordinate of the t-SNE. So how about the SWNE?
So that's a good question. I'll say we never really tried, but in theory it should be possible because, again, each of these coordinates sort of corresponds to sort of a gene loading. So you could basically, I'll say right now we don't have any functions for that, but it's theoretically sort of possible because you could basically construct sort of a vector of gene scores for each of these components and see sort of like you would for like principal component loadings if you're trying to interpret your principal component. So it's theoretically possible. We haven't done it yet, but that's a great idea.
All right, thanks.
So I think
yeah, that's enough for me. Thank you.
Thanks for the question. So we got a couple questions in chat. So for the noise data set with multi-feature, I think this might be asking about, Don might be asking about using distances to classify clusters. And we haven't tried to implement sort of a classification method with SWNE yet, but that's definitely an interesting idea, and maybe you can follow up if I didn't interpret that question quite correctly. But it should be possible to use a classifier, although I would actually recommend if you want to classify cells to use sort of a higher dimensional space than just 2 dimensions, because I think that will capture really the data set a lot better. So Marcus asks if, since seeing as SWNE is less able to differentiate local structures than t-SNE in muddier data sets, would SWNE lose its ability to tell apart clusters? So yeah, that's a great question. So I'll kind of navigate back to maybe a good example. So this data set. So yeah, basically SWNE doesn't separate the cells as well as t-SNE. So I think there are cases where clusters are sort of overlapping more than they would be in t-SNE. We haven't seen an example of, and it could be theoretically possible. It's kind of hard to go through and benchmark this on all data sets, but I haven't yet seen an example where t-SNE was able to, let's say, like completely separate two clusters, and SWNE is just, completely unable to separate them. It just kind of ends up looking a little bit messier, where you have SWNE isn't really able to keep all of its points, all of its cells, like kind of clustered together really nicely in the same way that t-SNE UMAP are. So you'll have cells that are kind of bleeding over from one cluster to another. But yes, it's possible that SWNE can lose its ability to tell apart clusters. Maybe if it's a very borderline kind of case where t-SNE is able to really separate them well, or separate them kind of just barely, and then SWNE can't. So yeah, hopefully.
Does that answer your question, Marcus? Okay, so yeah.
Okay, thanks. So we have another question. Have you applied SWNE on multimodal data with surface proteins? Because the landmarks would be very helpful to view all models on the same coordinate. We haven't, and that's also a great idea, and that's something that we've been thinking about as well, because yeah, I think there are all these multimodal methods now, and having everything on the same plot would be helpful. That would require a bit more algorithmic development, and I think there are some methods out there that do simultaneous kind of NMF decomposition of multimodal data. And there are other simpler hacks we could try before that too, but that's probably the way we would go. But we haven't tried that, but it's something we definitely want to try. And it's answer your question?
Does that answer your question, Nhan? We can unmute you so that you can directly chat with Ian. Okay. One more question from Tian.
Okay. So yeah, this is a good point. So I'll just read it out loud. So Tan mentions, so if you, for this data set, if you draw sort of a red box around on the SWNE plot that covers astrocytes and inhibitory neurons, you'll see that those two cell types are close together on the SWNE plot, but far on the t-SNE plot. So that's astrocytes and inhibitory neurons. Is this Tan? Is this plot, or is it the chromatin accessibility plot?
This plot.
Okay. Okay. Astrocytes. Oh, I see. Yeah. So maybe I'll present here. So here, I think is what you're talking about, and then they're kind of far apart on they're kind of basically across the map from each other on the t-SNE plot. Yeah. So yeah, no, that's a good point. The main thing that we kind of wanted to point out, yeah. So SWNE, and I would say this for UMAP as well, it's sort of really hard to orient clusters when you have this many clusters so that they're all kind of perfectly spaced in a way that's perfectly interpretable. So SWNE definitely, it definitely misses out on some global structure. But I would say that overall, so if you look at astrocytes and IN4, having this sort of white space between them, I would argue is at least sort of significant, whereas t-SNE, like, there's kind of a reason t-SNE just doesn't have a lot of white space on the plot, and that's just because all the distances tend to be compressed. So yes, that's true in this case, and I think you could probably point out a lot of other counterexamples to the main point we're making, but I would kind of just go through and mention that I think globally still that SWNE is able to capture more of the global structure, and yeah, but yeah, for that example, I think you're right.
Yeah, I agree that the global structure of the SWNE plot is great, but just want to say that the pattern of the three plots here is the they just want to separate the neuronal cells and non-neuronal cells.
Right, right.
Yeah.
Oh yeah, so this box, yeah, this is from one of the figures where we kind of did something else as sort of a demonstration. So that's actually not really related to this,
Yeah.
but yeah, sorry about that. That I should have removed that. But if you take a look at the paper, you can see we basically, for the neuronal cells, we're able to project cells from like a totally different type of data set using SWNE. But,
yeah, I think that's the right concept.
Yeah.
Thanks.
Yeah. Does that answer your question?
Yeah, I satisfied.
Okay, thank you.
Is there any other questions, guys? It seems like there is no question anymore, but if you do have any questions, please send an email to us for the question. I will list our email address over here, and so that we can directly send the questions to Yan and get the answers for you. All right? So after this webinar, we will make the recording publicly available on Bioturing's channel, YouTube channel, and on Bioturing's website. And we will also send the slides and the recording to your email info. And yeah, I would like to thank you. Thanks, Yan, for your valuable sharing. And thank you all for joining us. Have a good day and see you on the next webinars.
Thank you, Jenny.
Okay, thank you.
Thanks everyone for attending.